Greetings to all of you. In this video lecture, I will be teaching you about lipoprotein metabolism, which is a part of lipid metabolism. If we look into this whiteboard content, then I have shown you the different contents of the lipid metabolism. I've divided the whole content in 10 parameters. So the first one is the digestion, absorption, and transportation of dietary lipids. The second is lipoprotein metabolism, and so on. Like we have got triglyceride metabolism, then fatty acid oxidation, biosynthesis of fatty acids, cholesterol metabolism, eicosanoid metabolism, lipid profile test, kidney body synthesis, ketone body synthesis, and then we have fatty liver. So they are the subtopics of this large content, that is the lipid metabolism. In this video lecture, I will be only teaching you about lipoprotein metabolism. This is the content which I will be taking you in this video, and rest of the contents I will be taking you in another videos. So quickly we will shift to lipoprotein metabolism. So here we go. The lipoproteins. The lipoproteins are the substances which are water soluble complex of proteins and lipids. So they are complex formed by two different substances, that is proteins and lipids. The proteins are hydrophilic apoproteins, and they are of mainly four types: A, B, C, and E. So they are termed as apoprotein A, apoprotein B, apoprotein C, and apoprotein E. All of them structurally differ from each other. The lipid, which are hydrophobic, are actually triglycerides and cholesterol. The triglycerides are made up of fatty acids when they combine with glycerol. So the triglycerides and cholesterol, they are the main lipid components in the lipoproteins. So when this lipid combines with the proteins, it gives rise to lipoproteins. The lipoprotein are the dietary components of our food. If we see the structure of this lipoprotein, a common lipoprotein, which can be any type, it has got two parts: the central core as well as the peripheral covering. The central core consists of triglycerides and cholesterol, while the peripheral covering of this lipoprotein consists of proteins and phospholipids. The proteins are of different types, like ApoB, ApoE, ApoA, and etc. So this is the common structure in which the central core is formed by the triglycerides and cholesterol or cholesterol esters, while the covering is formed by the apoproteins and phospholipids. The lipoproteins in our dietary food diet consist of mainly four types of lipoproteins: chylomicrons, very low density lipids, intermediate density lipids, low density lipids, and high density lipids. So they are the different types of lipoproteins in our food. These chylomicrons consist of apoprotein A, apoprotein B48, apoprotein C, and E. And these are the different, this graph is actually showing the different components in different ratios. If we enlarge this graph, then we will find something else, something here. Just wait. All right. So VLDL, it is consists of B100 as compared to B48, C, and E. Apoprotein B100 and E is a part of IDL. LDL have got apoprotein B100 only, while HDL have got apoprotein A and C in its structure. So let us talk about these lipoproteins one by one and how they are metabolized. First of all, we will start with the chylomicrons. So they are small uh, spherical structures. They are synthesized in the small intestine after we intake the food, and these chylomicrons mainly consist of triglycerides as well as cholesterol esters. So the central core is always formed by the triglyceride and cholesterol esters, and the peripheral covering is formed by the apoprotein. Different types of apoproteins are there: apoprotein E, apoprotein A, apoprotein A4, B48, and apoprotein C. These are different, and along with the phospholipids. Now, these chylomicrons, it transport dietary lipids from the intestine to the peripheral tissue like muscles or adipose tissue. So they are the main transporters. If we talk about the metabolism of these chylomicrons, how these phospholipids found in these chylomicrons are metabolized in different parts of the body. So if we see this flow chart, it has got certain numbers: 1, 2, 3, 4, 5, 6, like that. Now what happens, we'll start with each number and I will be explaining you what is happening at this for the sake of ease. When we eat food, the food is digested and the chylomicrons developed in the small intestine. These chylomicrons are taken up by the lymphatic system. So via lymphatic system, they reach to the bloodstream, and through the blood they move to the different components, different parts of the body. So the chylomicrons reach to the lymphatic system and then to the blood. So when they reach to the blood, the fresh chylomicrons, it is called as nascent chylomicrons. In the center, it has got triglyceride, and the periphery, it has got certain proteins, apoproteins. So initially, it has got apoprotein A and apoprotein B48. But when it reaches to the blood, it interact with HDL, which is released or which has been formed by the liver. Now this HDL, high density lipid, have got apoprotein C second and apoprotein E. These two apoproteins are transferred from HDL to the nascent chylomicrons. Now this chylomicrons then get converted into a mature chylomicron, which consists of apoprotein E, apoprotein C second received from the HDL, and B48 and A are already present. And this is phospholipid in the center, triglyceride in the center, and it has got cholesterol at one corner. So the mature chylomicrons then go for the further processings. Now this apo C second, when this chylomicron reach to the body tissue like muscles or adipose tissue, in the walls there is a lipoprotein lipase enzyme in the walls of the tissue. This apoprotein C second activates this enzyme. So this enzyme actually converts the triglycerides and cholesterol, especially the triglycerides, into glycerol and fatty acid. It degrades it. So this is the metabolism of these triglycerides. So the fatty acids which are formed in the body tissue enter into the body tissue and then they are further metabolized like through the beta oxidation power. And this glycerol which is formed around the body tissue goes to the liver again and then it will be a part of the glycolytic cycle. The mature chylomicrons, when most of its triglycerides has been metabolized, it becomes small in size. And now the apoprotein C second and apoprotein E will go back to the HDL again. Now this small sized chylomicron with only B48 and apoprotein E and little amount of triglycerides and cholesterol ester is called as chylomicron remnant, and it is also called as intermediate density lipids, as I have shown you in this diagram. Intermediate density lipids, IDL, this one. So this IDL, which are chylomicron remnants, it goes very close to the liver and then they fuse with the receptors present in the liver and they enter into the liver. Inside the liver, the further metabolism of this triglycerides and cholesterol takes place, which are later on given to the HDL also. So this flow chart is there, and then I have shown you the write up of this flow chart. Say for example, if I have numbered it 1 here, So the step 1 will be written here. The step 1 is chylomicron synthesized in small intestine is released into the blood via lymph along with apoprotein A and apoprotein B48 on the surface. So this is all the write up. I'm not going to read all this. If you will read all these steps, that it will correlate with the flow chart. It will correlate with this flow chart. All right. So there are 5 or 6 steps I have mentioned here. So this is all about the metabolism of chylomicrons. And now next is VLDL and LDL. So we will see the metabolism. How does the metabolism of this VLDL and LDL takes place? VLDL is very low density lipids and LDL is low density lipids. Both are dealt in a at the same time. So if you see this diagram, here we notice that in the center there is core of triglycerides and cholesterol esters, and in the outer peripheral we have got certain proteins along with the phospholipids. VLDL, IDL, and LDL. I have shown you the three diagrams, and I will be comparing you. This VLDL have got B100 along with apoprotein A and apoprotein C second. The LDL have got apoprotein B100 and apoprotein E only. There is no apoprotein C second here initially. Then LDL have got apoprotein B100 and there is no other further protein over here, all phospholipids. So this is the basic difference between VLDL, IDL and LDL. They are all phospholipids. Now we are only concerned with these two here, but this third one is also connected. All the three are being metabolized in a similar way. Now flow chart of VLDL and LDL metabolism. If we look into this flow chart again, so we'll start with this number 1. So initially the nascent VLDL phospholipid is formed, which have got central triglycerides and cholesterol in the center, and the periphery have got certain proteins in it. So the VLDL, nascent VLDL or initial VLDLs are being formed from the liver. So the moment they are formed from the liver, they are transported into the from the liver it go to the blood, and the nascent VLDL have got apoprotein E, B100 and apoprotein C. Initially they have got all these three apoproteins. They also receive apoprotein C second and apoprotein E from the HDL synthesized by the liver. So these two apoprotein proteins are received by the HDL. Initially they are also having, but they also receive some of the apoprotein from the HDL also, that like apoprotein C and apoprotein E. So this nascent VLDL then becomes mature VLDL by receiving these two proteins. So if you see the structure of this mature VLDL, we have got triglycerides in the center, cholesterol in the center, cholesterol is here, and in the periphery we have got B100, ApoC second, ApoE. They are the different components of the apoprotein which a mature VLDL have got. Now this mature VLDL have got ApoC second, which activates the lipoprotein lipase, which is a part of the membranes of the tissues like muscles or adipose tissue. When this VLDL go to the adipose tissue, the ApoC second activates the lipoprotein lipase enzyme, which eventually converts the triglycerides and cholesterol, this triglycerides and cholesterol of the VLDL into glycerol as well as fatty acid. These fatty acids enter into the tissue and then further metabolized. The glycerol formed here goes to the liver, and it becomes a part of the different cycles like glycolytic cycle. Now, when the triglycerides and cholesterol of this VLDL are metabolized or they are being digested over here by the enzyme, this VLDL becomes small in size. Now it is called as IDL or VLDL remnant. This ideal even VLDL remnant also loses its fatty acids, uh, and this fatty acid enters into the body tissue. And now when the further more fatty acids are being released out from here, triglycerides are being released out from here. These ideal get converted into low density lipids. These low density lipids have got only B100 as its uh, peripheral protein. This apoprotein E is also lost from here. These LDL are then received by the receptors present on the wall of the liver, and then these LDL enter into the liver. The furthermore triglycerides which is present in this LDL is metabolized into fatty acids and cholesterol inside the liver. This fatty acid combines with the cholesterol inside the liver and becomes triglyceride again, which is released into the HDL. Okay, now these LDL also release certain cholesterol to the body tissue here. From here, the cholesterol is also released into the body tissue. So this is the flow chart. If I will show you again, the cycle goes like this. VLDL are formed, get converted into VLDL, get converted into mature VLDL by taking the ApoC second and ApoE from the HDL, and then these ApoC second and ApoE from this mature LDL goes back to the HDL, and these mature VLDL they digest the lipoprotein that they digest the triglyceride with the help of lipoprotein lipase, and then this structure VLDL becomes small in size, which is called as VLDL remnant or IDL, which get converted into LDL, and from this IDL and LDL the fatty acids or cholesterol also exit into the body tissue, and LDL further finally formed is taken up by the liver, and then inside the liver it is further metabolized. So like this the metabolism of VLDL and LDL takes place. So this is the write up of this flowchart. I'm not going to read everything here. Now metabolism of HDL, high density lipid, how does it happen? If you see this flowchart here again, if you see this flowchart, I'm going to explain you the metabolism of high density lipids. So we'll start with this here, number one, the nascent HDL. Initially, the nascent HDL is formed from two places. The small intestine gives rise to nascent high density lipid, and it is also synthesized from the liver. So in the center there is cholesterol as well as triglycerides, but it has got ApoE, ApoC second, ApoA, and there is an enzyme LCAT. I will be showing you the full form of this LCAT later on. What the role of this LCAT enzyme? It is also one of the protein present in the wall. So this nascent LDL, when it receives cholesterol from VLDL and chylomicrons, so it in, it bulges and becomes LDH3, and this enzyme actually catalyzes this reaction. I'll be explaining you this reaction in a short while from now. So this nascent HDL is converted into LDH3 when it receives further cholesterol, and then cholesterol is further metabolized by the enzyme LCAT. Now this HDL gives cholesterol esters to the VLDL, and the VLDL in turn gives more of the triglyceride back to the LDH3. So now this have got triglycerides, cholesterol esters and cholesterol inside it, which is given to the liver when these LHD fuse with the liver. When this LDH3 go to the liver, it is received by certain receptors called as hepatic LDH receptors or scavenger receptors B1. These receptors receive this LDH3, and then this LDH3 enters into the liver. Inside the liver, these LHD are further metabolized as cholesterol esters, and the cholesterol esters of these LDH3 is released from the liver as bile salts like bile acids and bile cholesterol. All right. So, what is the role of this LCAT? This LCAT is actually lecithin cholesterol acetyl transaminase. All right. So, this enzyme lecithin cholesterol acetyl transaminase, that is LCAT, catalyze this function. If you see this diagram here, The cholesterol is converted into cholesterol ester here with the help of this enzyme LCAT. The cholesterol is being converted into cholesterol ester. And how does it happen? The fatty acid of the lecithin molecule is combined to the cholesterol, this squarish structure combined to the cholesterol, so it becomes like this here. So the lecithin, the fatty acid from the lecithin is transferred to the cholesterol, so the cholesterol with the help of this enzyme get converted into cholesterol ester. This is the structure of the cholesterol ester, and this lecithin is converted into lysolecithin by losing its one fatty acid. It becomes lysolecithin. So this reaction has been catalyzed by LCAT. So here we have seen that this LCAT is converting the cholesterol received from the VLDL by the nascent HDL into cholesterol esters. So when the cholesterol esters are present in the nascent HDL, this nascent HDL get converted into LDH or mature LDH. HDH, sorry, here I have written HDH. It has to be HDH, high density lipids. All right. So there are two important concepts which are present here. Sometimes the cholesterol accumulates in the body. It is because of the absence of this enzyme LCAT. Now, this phenomena where it reverse cholesterol transport occurs, it is a process of transport of cholesterol from body tissue to liver, so that the excessive cholesterol is removed from the body tissue, and it is done by the high density lipids, because it is transferring the cholesterol from the body tissue to the liver, and the liver then further metabolize it. If we see that HDL have got LCAT, this LCAT level blocks, it is absence, then this cycle reverse cholesterol transport will be blocked. If this reverse cholesterol transport is blocked, so there will be accumulation of cholesterol in the body, so it will lead into the high level of cholesterol. That's it for this video. Hope you have understood the concepts of lipid metabolism, especially lipoprotein metabolism. Thank you very much.